Oh, okay. So hi, good morning, good afternoon, everybody. We're a bit early for the webinar, so we will wait 2 or 3 minutes before we get started. But already, let's say thank you for joining. So we're at the top of the hour, but I still see people joining. So we'll start in 1 or 2 minutes. Okay, so I will get started. So just a couple of little bits of information for yourself. So on the right hand of your screen, there should be a chat icon where you can post questions during the webinar. But then there is also a Q&A icon. So that's a bubble with a question mark in there if you want to also post a question privately. So I'll run through the webinar today, and then towards the end, I'll try to address as many of the questions that I can after the webinar. But if I'm unable to answer those questions immediately, they'll be saved, and then we will then respond to you via email. So good morning and good afternoon, everybody. And first of all, thank you for joining the webinar. And we would like to describe to you and demonstrate to you how at Encardia we're using human iPSC derived models and how they are then a robust screening solutions to support various drug discovery endeavors. And ultimately, at Encardia, we're a big believer of that. To bridge this translational gap that we see in drug discovery, we need to use human biology, not only to inform ourselves, but help drive decision making to increase the success of therapeutics getting to patients. And to introduce myself, my name is Shushant Jain. I'm the Director of Drug Discovery at Encardia. My expertise is primarily neuroscience, but since moving to Encardia, also learning quite a bit about cardiac and other types of diseases. So briefly, the agenda is introductions and challenges in the drug discovery space, and in general, I'm trying to help understand why there are so many drugs that are failing, and how we believe iPSC models can actually help that entire drug discovery process. So then we will go over Encardia's iPSC platform, both from a reprogramming primary cells to iPSCs, all the way through to differentiation. And then ultimately how we've used those differentiated cells in both the cardiac, neuroscience, and also in the safety space. And as I mentioned earlier, we will then also have a conclusions and a Q&A. So drug discovery. So to identify therapies which are both clinically efficacious and safe is a very challenging endeavor. And currently, there is a big disconnect between early discovery and clinical success. With I believe over 90% of drugs failing at various points throughout the entire process. And on the left here, what we see are three kind of areas where we really believe that using iPSC-derived models can really be beneficial. So first of all, in the target discovery and validation space, only approximately 10% of targets identified through the traditional target-based tools, only 10% of them have actually led to clinical success. So we need to be able to use models and use tools which are much more informative about whether the target is relevant for disease and if modulation of that target is really going to have therapeutic benefit. We've also seen there are translational challenges. So what the field commonly terms as translational value of death. So we need to have more complex, more holistic models of human disease to understand if the therapy is actually going to be effective. And then ultimately stratifying the patient population that you are targeting with these therapies. So we know that patient populations are not uniform. There is significant both clinical and pathophysiological heterogeneity, leading to significant variability in clinical trial outcomes in terms of responders and not responders. And ultimately, it's a significant reason, or leads to a significant number of potential therapies failing clinical trials. So the question begs is how do we actually fill this gap? So on the right here, what we see is that using phenotypic human disease models, this is really bringing us closer to the human biology. In combination with omic technologies, which give us a deeper understanding of the molecular biology behind the disease, but also if target modulation can actually restore the entire cellular system. And then in combination with AI and machine learning, help us understand, interpret that data and integrate that data into a more effective drug discovery strategy and actually a more human-centered drug discovery framework. So why do we want to use iPSCs for drug discovery? So first of all, they're self-renewing and can be differentiated into virtually any cell type in the body. This is in contrary to, for example, primary cells, where sometimes they can be difficult to source, and this is particularly true for cardiac and for CNS cell types. And there is significant patient-to-patient variability associated with those primary cells. The iPSCs themselves, they retain the patient-specific genetics. So if we're looking at Mendelian diseases, you can maintain that mutation. But also if we go to these more complex diseases where there are polygenic genetic risk factors, these patient-derived iPSCs retain that genetic component. They retain human biology, as you would expect, and therefore have the human pathophysiology. So, for example, when it comes to, my background is neuroscience, as I mentioned earlier. So when you're looking to model certain diseases or look at structures such as synaptic function, you either have to use a rodent model or you have to begin using a human iPSC-derived cells. And then fundamentally, iPSC technology is still, I would say, quite young. So since its discovery in 2006 and 2007, the technology has advanced significantly. So, and I'll come into this in my next slides in a bit more detail. So in the early days, culturing and differentiating iPSCs were a bit of a pain, to say the least. But now solutions have been optimized through, various companies, but also academia, that have actually scaled this technology to ensure that we can begin to produce billions of cells, both robustly and reproducibly. So this is what I was mentioning in my last point from the previous slide, is that when iPSC technology was discovered and was being adopted across various both academic and biotech companies, there were significant issues with it. So for example, there was a low efficiency of reprogramming. So this is when you convert a primary cell type into the iPSC. There was significant genetic instability. So there was lots of chromosomal aberrations which were accumulating in these iPSC lines. The differentiation from the iPSC line to the terminal cell type was quite often heterogeneous. So although the basic protocols were there, quite often the purity of the population that was produced was quite mixed, and these protocols were challenging, to say the least. They were long, they were complex, quite laborious, and quite error-prone. And then ultimately, the cells that were produced from those protocols were generally very immature and almost fetal-like. So for a lot of human diseases which are age-related, this created the problem, or did not solve the problem of having a good enough model for that disease. So as I mentioned, the field has advanced quite rapidly in a number of different ways. So now there are quite optimized, and at least at Incardia, there are optimized methods for reprogramming primary cells from various patient populations into iPSCs. So the protocols themselves are very standardized, allowing us to create iPSC cell banks. There are robust QC procedures in place. So this is really, I think, initiatives which have been driven by the ISCCR, where they are looking at, deep characterization of those iPSCs to ensure that they are, genetically stable, they express the correct markers, and are indeed true iPSC lines. There have been a number of different initiatives to try and scale the protocols for differentiating patient iPSC lines into that terminal cell type. So at Incardia, what we've employed are using scalable bioreactors, which are in combination with chemically defined differentiation medias, automation of the protocols, and 24/7 in-process monitoring. This allows us to produce a large number of a very pure cell type, and we're able to do this both robustly and reproducibly. And then ultimately, we have the maturation question. So there are efforts, certainly in Encardia, to indeed have that differentiation in 3D to enhance that maturation of those terminal cell types, but also beginning to bring these cell types together, so in a multicellular structure, again, enhancing the maturity of that overall model system. So in Encardia, we, like I mentioned, we have quite a standard process for onboarding or accepting an iPSC line or receiving primary cell types and reprogramming them into iPSCs. So once the iPSCs have been qualified and have been banked, they then go through a process definition. So this is a small-scale bioreactor where we optimize and validate that the differentiation protocol to get to your terminal cell type is working, and if required, we can, tweak and improve both the media composition, the timing of addition of key components. And this is actually, depending upon the differentiation process, quite a robust platform to ensure that when you begin to scale up iPSC differentiation, we are more confident that we are going to get the cell type that you require. And then we have post kind of that process development. We then have pilot validation or an industrial scale, and this is very much dependent upon your needs. So at the pilot validation, we produce between 25 to 500 million cells. Normally, these are sufficient for initial experiments, perhaps proof of concepts. But then if you really want to support end-to-end, a large-scale drug discovery program, we can start scaling up the differentiation into the billions so that you don't have to continually do lot-to-lot validation in your assays throughout that entire process. You simply produce the cells once, you qualify them in your assays, and then you have sufficient cells for your remaining runway. So also in Encardia, we differentiating to this cell type is obviously that's important, but it doesn't stop there. So having a cell which expresses the cell type marker is indeed very important. But along with that, we want to ensure that the cells themselves are behaving as you would expect in the human body. So the example I'm showing here are for our N-site cardiomyocytes. So let me just get the pointer. So on the left, this is our typical, or part of our QC procedure for our cardiomyocytes. So we look for the percentage of troponin T positive cells. And what you can see is that across a number of different batches, we have quite a high purity, so over nearly 90% purity of troponin T positive cardiomyocytes. And when we look at them underneath the microscope and stained for sarcomeric structures, we see that this typical patterning within these cardiomyocytes. So coupled to that is, this is primarily cell identity, but we also want to ensure functionality. So this is electrophysiology readout, so using multi-electrode array of our iPSC derived cardiomyocytes that show stable baseline field potential durations and reproducible waveform morphologies. And then they also respond to expected pharmacology, or they respond as expected to different pharmacology. So when treatments with compounds such as nifedipine, vidofetalite, we see the expected changes in the field potential duration in a dose-dependent manner. Going back to what I said earlier, supporting you from end to end for your drug discovery process. So depending upon your drug modality, but also where you begin, this entire drug discovery process can be lengthy. So it can be from 3 to 5 years. So the question that this slide is trying to answer is, are the cell types stable over time, in that if you produce sufficient cells in year 1 of your drug discovery program, can you still use them in year 5? So what we have here is we see that across different batches, as I mentioned earlier, the viability of these cardiomyocytes is very high. So again, over 90% viable, but this viability does not drop over time. So what we have here on this bottom right-hand graph is that these are the cells thawed when they were first produced, and then 5 years later, from the same batch, we have thawed those same vials. So what we can see is that there is very little change in the viability of these cardiomyocytes. In terms of the consistency, what I mean by consistency is the cellular identity. Again, across the different batches, we see that the percentage of troponin T positive cells remains very high. And again, if we look on the graph on the bottom left, what we can see is that over time, the percentage of, there is very little change in the percentage of troponin T positive cells. And it's very similar, again, story from batch to batch in terms of the expression profile. So overall, the cells that are produced are stable over time, eliminating the need for revalidation cycles. So ultimately, this is one aspect of a very important aspect of drug discovery. So really pushing and generating a human relevant cell type. But at the moment, it's only a cell type, but we need to be able to use those cell types in an assay, in a readout that is able to enhance decision making, but also enhance, or should we say, bridge that gap, translational gap that we see in the drug discovery process. So this is just a snippet from a really nice review from 2015, where the authors try to define what they, in their opinion, what they think, and also in my opinion, what they think is perhaps the most highly relevant assays and readouts to try and bridge this translational gap. So I won't read through every bit on this slide, but for example, in terms of a system, so this is really a cellular system. Then we have the stimulus and the readout. So if we stick with the system, in their opinion, the lowest relevance is a cell-based system with a signal protein which has been overexpressed. So this is, and the cell line they use here, for example, is a HEK3. So we know that the HEK3 is not really relevant, even though it's not really relevant of human biology. There are significant karyotypic abnormalities and chromosomal abnormalities. And ultimately, how are you going to translate data from that cell-based system all the way into a complex human cell type? And then on the other end is the highest relevant system. So these are iPSC derived cells. So these are really, genetically normal. They have been differentiated to the correct cell type. Sometimes, if needed, if you're modeling a disease, have that patient mutation. And in parallel to, that the cell type that you want, these systems can then be used in a multicellular model to, again, start improving or enhancing that translation of that model. In a perfect world, we would need no stimulus to induce that disease phenotype. So really, the genetics behind the disease is really sufficient to produce that disease phenotype. And then the readout, really as close as possible you can get to the disease manifestation. So really something that will give you confidence that if you manipulate a target and ultimately a pathway or set of pathways, can you rescue that functional deficit that we observe in humans? An example of this would be, say, modulating a target gene expression. So that is very distal to the clinical endpoint. Whereas the more functional manifestation, so can you, in a cardiomyopathy, for example, can you actually rescue the arrhythmia and so really rescue the function of the cardiac cells? Again, building confidence that your therapy is able to modulate the disease itself. So pivoting from that, I'll present some case studies where we have performed some screening in the cardiac space in the CNS, and really how these models have helped clients make decisions and allow us to model diseases which otherwise would not be possible using other in vitro model systems. So the example here, this is a cardiac safety study that we ran for close to a year, where the client compounds induced cardiac arrhythmias, and standard in vitro assays were unable to pick up this toxic effect. So in this scenario, what we did is that we qualified a lot of cardiomyocytes for a very specific assay setup for this client, in being able to pick up this this compound-induced cardiotoxicity, in particular inducing these arrhythmias in this in vitro system. And then kind of, after reserving that lot, what we were able to do is support that client, as I mentioned, for close to a year, where the chemists were modifying the backbone of these molecules. And this is the initial molecule that they provided to Incardia, where in a dose-dependent manner, we saw significant decreases in beat rate induced by that compound. Whereas after the chemists had done their magic, even at the highest concentrations of their molecule, we could not detect any arrhythmias. Again, that gave confidence to the client that they were able to move forward to the next steps of their program. The other aspect, and I touched on this briefly, in that iPSC derived cells, one challenge here is the maturity. So there are multiple different paths that are being taken to try and enhance the maturity of these iPSC derived cells. And this is an example of what we've done at Incardia. And this was in collaboration with Richard Davis's group, at the LUMC. Where we've taken 3 different cell types. So this is ventricular cardiomyocytes, endothelial cells, and cardiac fibroblasts. And we combine them using automation to form this microtissue. So this is a 3D cardiac microtissue, again, composed of these cell types in a very specific ratio. They are then maintained in culture for up to 21 days. Again, this 3D structure with the multicellular composition, again, increases their maturity. And then we're able to perform a number of different assays with these 3D cardiac microtissues. And the example I'll go into is calcium imaging with this model. So one aspect is that there's a disease, a cardiac disease called CPVT, where in humans, if there is a lot of stress on the heart, so this could be exercise, for example, the individual suffers cardiac arrhythmias and potentially have fatal consequences. At least in a 2D system, this CPVT mutant line was not showing really robust cardiac arrhythmias. So as I mentioned, in collaboration with the LUMC, after forming these 3D cardiac microtissues and the cardiomyocytes were produced from a CPVT mutant patient, we could see that upon stress of these cardiac microtissues, we were able to induce cardiac arrhythmias. And then with the compound flecainide, we were able to rescue this disease phenotype. So, and actually flecainide is the treatment for patients. And ultimately, the goal of these kind of approaches is to help stratify patients. So we know that in the clinic, there are patients who will respond, for example, very well to flecainide, but there will be patients who do not respond or do not respond as well to that dosage of flecainide. So if we're able to build a platform where we can begin taking patient-derived samples, converting them to iPSCs, and actually performing a screen to identify who is a responder, non-responder, and at which doses, and potentially identifying molecules or other therapies which may help those non-responders to flecainide. So in that space, again, as mentioned, together with the LUMC, we performed a compound screen using the SPECS repurposing library. So this is these are compounds that have at least gone through at least a phase 1 clinical trial, but for whatever reasons have not progressed any further. And using this fully automated workflow from microtissue formation to compound addition and then to finally to assay readout, we were able to screen all these 2,000 compounds in triplicate within about 2 days. So overall, the Z factor was around 0.6. So Z factor is a measure of screen quality. So this is quite high. And ultimately, we were able to identify a number of potential not novel compounds, but new compounds that could be potentially repurposed for the treatment of CPVT. So moving into the CNS space, so very similar story to how we worked with the cardiac, in that we produced a large quantity of neurons in this instance, again using the bioreactor technology, and then we can do our standard QC, so really verify the cell type identity. But into this, we can also then plug in both electrophysiology readouts as well as other molecular readouts to really ensure that the model itself is fit for purpose for the drug discovery, for your drug discovery program. And ultimately, automate the entire workflow, safe, again, very similar to the cardiac, from cell seeding to media refreshes, compound addition, and then assay readout. And overall, this leads to very significant or highly reproducible data from biological replicate experiments. So coupled to that, the workflow for screening or for hit identification as well as potency, it's important to not only understand if your compounds are doing what you want them to do, so engage the target or modulate the target. It's also important to understand if the therapies that you're working are also safe. And safety is a big thing to ensure that the sooner you can detect any potential issues with those candidates, the quicker you're able to begin working on that problem. So in coupled to the efficacy part of your study, we can also begin incorporating safety aspects. So the example we have here is a relatively simple calcium assay, and this is again in the CNS space, where we seed our neurons in the co-culture with the astrocytes, and then after they have matured, so again going back to this need for maturation, so in fetal-like neurons don't show spontaneous calcium activity. You really need those mature neurons. And as they mature, they have this spontaneous calcium transients. And we quantify various aspects associated with this spontaneous calcium activity, such as peak height, peak-to-peak rate, as well as something called the calcium oscillation score, which we'll come to in a second. And ultimately, what this allows us to do is begin to screen for potential toxicities associated with your molecules. And the example I'm using here is from ASOs. So in a paper by Hagedorn et al., they tried to correlate in vitro using primary rodent neurons. Actually, they treated them with ASOs, and they correlated the effect on spontaneous calcium activity with the potential neural toxicity phenotype that they had observed in vivo. And what they saw was quite a remarkable correlation, that if the ASO was toxic in the animal, it's caused significant disruption of this spontaneous calcium activity. So what we did in Encardia is bring that away completely from the rodent system to the human system. So using a human neuronal and human astrocyte co-culture, and again ensuring that the protocols we have in place get them to the maturity level that we need, such that they have this spontaneous calcium activity. And then when we treated them with toxic ASOs, we could see that the spontaneous calcium activity has almost been completely reduced, whereas a non-toxic ASO shows, a small blip after milliseconds after the addition of the ASO, but very quickly there is virtually no difference in the calcium profile prior to addition of the ASO. Excuse me. And this kind of leads to this type of data, where example for ASL1 has very little differences or very little changes of in calcium oscillation score. So the calcium oscillation score is just an aggregated measure of the peak rate and peak amplitude. So at the lower concentrations, it has virtually no effect on calcium activity, whereas at the higher dose, there is quite a significant impact very quickly after addition. But what we see is that by the end time point of readout, so 10 minutes post-addition, we can see that the calcium oscillations have almost recovered back up to only about 30–40% below the maximum. ASO2 at the higher dose has quite a significant impact on calcium oscillations, where at the lower dose it has a minimal impact on the oscillation score, indicating a narrow safety window, whereas ASO3 has quite a significant impact on calcium activity, highlighting that this molecule is likely to be toxic in vivo. So kind of for me, it's at least a very natural workflow to be able to go from screening in robust human models to identify your hits, and then coupling them to human models for understanding if you could potentially see any toxicity, allowing you to make decisions much quicker in terms of which molecules you can progress to the next stage with more confidence. So, to summarize the key takeaways, so iPSCs and really, bringing the human biology is critical in bridging this translational gap in drug discovery. I think at virtually all stages of that drug discovery pipeline. We've re-summarized the key benefits associated with the human iPSC models, and in particular the expertise that we have in Incardia to be able to support you with each of these efforts, and in particular being able to be flexible in how we can get you to, as quickly as possible and with as much confidence possible, hits that are going to be safe and translate to the patient. So on that note, I will stop and look at the questions. So I have a question here. So is the full characterization of the non-cardiomyocytes in the culture system always available? Yes. So overall, the purity of the cardiomyocytes that we produce is extremely high. So we're looking at above 90%. From the non-cardiac myocyte population, we do include markers for other cell types. So for example, fibroblasts in there, just to understand what percentage they are in there. But also for the other cell types we always try and produce include markers, which will help us to clearly define the population of cells that are going into a vial or going into an assay. Does that answer your question? So I have another question, and thank you for that. With respect to the patient-derived iPSCs, do they consistently maintain the mutation profile which caused the pathology in the first place across the process? Oh, that's a good question. So there will be, so at the iPSC stage, there will be mutations which are just affect viability, quite dramatically. So those are always going to be difficult to model. Most of the times, at least in the iPSC space, we see genetic stability. So the mutation does remain in there. If the mutation itself, if the mutation itself interferes with the differentiation process, then there could be a potential there's some sort of selection against that mutation. But in general, that would likely lead to collapse of that differentiation protocol. So ultimately, you will not have any cells available. But if the mutation doesn't affect differentiation, in general, we don't see any changes in its prevalence. You were interested in non-cardiomyocyte multicell. Yes, so so this goes back to the original question about the full characterization of non-cardiomyocytes in the culture system. In particular, they were referring to the 3D microtissue. So we have characterized them in terms of the non-cardiomyocyte cells in the microtissue, but the way we actually form the microtissue is very, very specific. So we do a pre-culture of all the 3 cell types. This allows us to eliminate dead cells or dying cells. And then when we form the microtissue, they go in a very, very specific ratio. So I don't remember the exact numbers off the top of my head. And ultimately, what we do see in the microtissue is that composition or that the planned composition of the microtissue remains the same. So there are no, when we form the microtissue, we're looking for dead cells or for a necrotic core. We don't see any of that. So essentially, the 3 cell types are maintained in the same ratio. Okay. I don't see any other questions coming through. Perhaps I'll then just jump to this last slide. If you questions always come up later, so if you think of anything that you did not have the opportunity to ask and want to reach out to us, you can visit our website, or you can email at info@incardi.com, and then we will happily provide you, if we can, with the answers that you need. So again, thank you for your time today. I much appreciate you making the effort to join, and I hope this was useful for you. Oh, I have one last question. Have you considered using mitochondria as a readout? So quite routinely, we look at mitochondrial dysfunction, more from a metabolic perspective in the 2D cardiomyocytes as a monoculture. Essentially, the 3D microtissues can also be amenable to high content imaging or plate reader-based readouts. So that is quite feasible to incorporate that into the model systems. Okay, thank you very much, everybody. Goodbye. Should I end?